Thank you all for joining us for the MPS seminar. And before we start, I just want to inform all the participants that the session will be recorded. And if there is anyone that don't agree, they should keep the camera off and write the questions on the chat. During the presentations, all the participants should keep the microphone mute and just turn it on to do the questions. So MPS NOVA is a twinning project led by Instituto de Tecnologia Química e Biológica, ITQB, in collaboration with Nova Medical School, both part of Nova University of Lisbon. MPS NOVA aims to establish an European hub of excellence in microphysiological systems. Fueled by pluripotent stem cells technology to strengthen Nova University research and innovation capacity in the areas of chronic disease and host microbiome interactions. MPS Nova-driven partnership of international leading institutions will enhance the existing multidisciplinary knowledge and expertise of Nova MPS Hub, advance our best practices in research and training, and expand collaborative networks. And if you want to know more about MPS, you can follow us on all our social media, LinkedIn and X. And you can also visit our webpage to know more about the project, the consortium, the workshops, exploratory projects, and all the events of MPS. And now I will present our first speaker, which is Krzysztof. I hope I pronounce it well. And Krzysztof obtained his PhD from University of Southern Denmark, has a strong background in biomedical science, having working within research for the past 25 years. He focused on advanced cell culture technologies and development of drug candidates, testing pipetting based on toxicology assays and in vitro models. He is the co-founder and CEO of CelVivo apps and extraordinary professor at Center of Excellence of Pharmaceutical Science, Faculty of Health in South Africa. And today he is going to talk about advanced organoids manufactured with ClinoStar technology. The floor is yours. I will stop sharing and you can start.
Okay, thank you very much for a very nice introduction. It's a great pleasure for me to talk to all of you. And today I would try to show you a bit more about ClinoStar technology. This is an technology which is based off our company, but actually it comes from my time at the universities, where we actually created it to basically help our own research in the lab. So, oh, I can see, yes. So we all are here because of a problem shown on this slide. I think everybody has seen this before, that the methods which we were using until today, they are good, but they are not always doing everything what we would like to be made with models where we model the human being. So we have animal models which are very holistic, but unfortunately we have a species differences. And then we have our in vitro models where so not so long ago they were based on the monolayer cultures, which were also maybe based on human cells, but not on the tissue-like organizations. And what I usually say, it's actually what we need right now is a farm. And I know that doesn't sound like, oh, that's what we need in the laboratory. But basically what I explain to my colleagues and students is that, what we do in our laboratories right now is more like this hunter-gathering society. Everybody needs to learn from the scratch, and everybody has his own secrets, and them actually hunting on means that the process starts to be a main driver, not the results. And this is why I say we are needing actually a farm. We need to more concentrate on the results, on the biological problems, not on the how we make our models. And I say we need the farms because farms actually describe the place where you can adapt and control the environment, where you can support larger populations, so means more experiments, and you can also support more complex and advanced societies and systems. So that's what I'm saying. What we are now doing in the 3D cell culture world is going to a farm-like philosophy. And of course, our farms need to be adaptable. So we all know that organism is a very complex construction, and we have a very different conditions in our organism. And this is what we also have to be able to adapt with our systems. And I also would say that we talked about this for a very long time, but actually only the 2022 and the FDA Modernization Act make a very, very big boost forward for all our three-dimensional cell culture systems, because this act made signed in 2022 replaced actually an original law from 1938, where the use of the animal experiment was mandatory for the for development of any new drugs. And actually, right now, it is not anymore a need. You can use other alternative methods. So, as it was mentioned before, I am actually right now a chief scientific officer in the company called CelVivo. And CelVivo, in CelVivo, we have we work under this concept, which was actually developed still when I was at the university. We call it RARE. So our model definitely should be relevant. Our model should be abundant, because one way is to create a super model, another one is to have it in the sufficient amount to actually perform all the experiments we wanted to. And of course, it has to be reproducible. And the last but not least, it has to be easy to use. Models should not occupy our scientific mind or mind of scientists. They should think about biological problems, and the models should serve their needs. So very quickly, many different 3D cell culture models have been created and are in use. We can divide them into the basically two big groups, static and dynamic models. With static models, we actually have a very nice and stable, mechanically stable environment. However, we are facing this problem of the passive diffusion, which creates diffusion depletion zones. And that actually limits the size and the complexity of the constructs we can create there. So people very quickly came out with the idea, okay, let's remove these diffusion depletion zones by basically moving media. So we went to the active systems, where we basically use a physical movement to create a media flow. So we remove the diffusion depletion zone. However, with this problem come a new problem, which is called shear force. And physiological shear forces exist in some organs, or some models are needed. For most of our models, we don't need it to, and we do not need definitely the technological shear force, which we are introducing with many systems. So there come our approach, where we actually utilize an ClinoStar idea to actually remove the diffusion zone without increasing the shear force. And how it works? We basically use a force of gravity and force of uplift created by a medium, so a liquid-like solution. And we create this rotation. So when you look here, it's a very, very dynamic. However, if I will remove the rotation of the system, so basically the camera is now rotating together with the rotating vessel. So now you can see what's happening inside the vessel. So I usually would say that's like the first picture was like if you would be watching a passing train when you stay on the railway station, and the train passes you, and then you think, okay, those passengers are moving very, very fast. But if you're inside the train, even the train is moving, and you look on your fellow passengers, they are quite comfortable and not moving so much. So that's the whole secret of the ClinoStar, that you actually don't have turbulences, and you have very stable diffusion. How we can measure it, or how we done this? It's actually here you have the same black picture which you have seen on the previous slide, but actually made in the numerical calculation with a software which actually counts or describes the liquid dynamic. And what you can see here is that all the trajectories are almost identical. So that means that all those objects, they have very identical conditions. And that's a secret for a reproducible culture, that you are able to create an reproducible conditions for them. And you can see this on the graph here, that actually most of our objects are having a very similar conditions. It's only a few outliers which are having a bit different conditions. So a bit more about this. So if we have our reactor and we are rotating it, as you have seen it before, and we have our object. So we have rotation. That's the uplift which we create. Then we have the inner rotation, which actually creates the diffusion through the object. And of course, we have also the rotation of the object by itself, created by the shear or by the fraction of this object and the surrounding medium. So you basically, by this, you have a very uniform distribution of those forces. So those are the forces which we have inside of our reactor. However, we also have to remember about another one, which is the gravity out of Earth. And I know that's what we have. We have existing in the reactor. So we actually published a paper in 2018 where we calculate all those forces, and then that's a lot of numbers and a lot of equations. I know that not all biology people like those physical calculations, so we have actually made something a bit simpler, and I would say something which you can relate to, and it's the feelings, like what this object feels. So we have those main movements, but I will replace the object with, say, I will personalize it. So that's an our construct, and what this construct feels, it actually feels that it's immersed in the liquid. It feels that it wants to go down, so that basically all the time falls down, and it's supported by upcoming liquid. And it's actually rotating. So what I would like to say here is that we have this relative feeling. The object actually does not feel that it's rotating. It's like we are not feeling that the Earth is rotating and we are walking on the rotating ball. The object actually feels only the down movement. So that's actually kind of important. But let's say what we have through this environment, we are able to support much bigger constructs that in the other approaches, where we have it's like a similar as to a whale. If you can imagine that the whales in the water can be much bigger organism than anything what walks on Earth in the standard conditions of the gas-based gravity. And then we have also a very nice distribution of diffusion through these rotations, and also a distribution of the gravity vectors, which allows us to actually have a development of the construct in all directions nicely. And yes, we do have around 1G because we are working on Earth, and we don't increase the speed to values which actually will hinder this number. And that's a very important thing, that this low shear environment support is actually could be also called a continuous laminar fall. So basically, it's all the time falls down, and that's unique for ClinoStars. And then very quickly, this is what how the ClinoStar, what I'm meaning about the ClinoStar. So of course you can find quite more information on our webpage, but basically this is a fully autonomic system where you have an climate chamber which controls temperature, CO2, oxygen, and humidity is controlled actually in the disposable reactors. And you have also a system based on the cloud, so you basically can control that unit from any internet-capable device. And that system is, of course, able to do conditions for different regions of the human body. And we have published quite a lot about that already. So I would say go for this webpage with the publications. What is important for us that we always try to have a relevance to in vivo. In old days, when I started, I would say people would say, let's compare 3D to 2D. That's not thing which we should do. We should compare our 3D to an in vivo situations. Two-dimensional or monolayers are the models, and are we as there or better? That it's nice to know, but what is important to know is actually our importance to in vivo situations. So very quickly, how we can work with the system. So when you have to create a construct which the ClinoStar will support as a live support system. So you can create them very simply with an just putting cells, single cells into vessels, or you can actually design them by force aggregation, or with use of the hydrogels, or actually also with the bioprinting. So very simple methods where you basically have your single cell suspension and you put them into the ClinoStar systems. And if those cells are able to produce and secrete even minute amounts of extracellular matrix, they will conglomerate, and then physics will take over, and then they would actually create those nice constructs. Here you have an examples from our work from the Northwest University in Potchefstroom, where we actually create a construct where we use them in the drug testing pipeline. So we need a lot of material for different tests, and you can see a very nicely created construct and with a very nice cellular composition. And I have to say those are the native constructs, so nothing is added except the cells to those. Here is a method. That's another method where we are actually using an force aggregation. So some of you may know Agrewell plate or Euplasia plates or Kugelmeyer plates. So those are micro-pattern plates where you can actually conglomerate cells. And then here, this is a very nice method if you, for example, would like to do a co-culture. So you conglomerate no numbers of a certain type of cells and basically grow them further. And here I could show you a glimpse of the results where we actually use this method to create a fibrotic liver model where we combine two types of cells and then grow them inside of the reactor. And you can see that we actually have deactivated the fibrotic, the stellate cells. And you can say, okay, maybe they have died out because the marker went very much down. But we actually have labeled them with the fluorescent labeling or tracker, and we can actually see that they are existing through the whole period of the growth. And of course, we have tried to use this system for the stimulation. So we use the TGF beta 1 to see if we can actually affect the system, and we can see that yes, we do are able to clearly distinguish the higher doses of TGF beta, and then we also can see that we have a very much differences in the collagen production and placement in those constructs. So that shows very nicely that the system actually is responsive and capable of showing physiologically relevant results. So yet another example of the work where we actually create and design constructs with use of hydrogels. And with these systems, we can do it on the hydrophobic surfaces, or we do it directly into the reactor. And you can see how nicely they grow further. And I would say that again, here you can see another part of our experiment with the fibrotic livers, where we actually were studying the reaction of the stellate cells or fibroblast-like cells on treatment, treatment with a fibrotic agent and with the antifibrotic drug. And then, first of all, when we use our system, we are able to deactivate the cells. So basically, they are reaching much more stable conditions, non-activated conditions. We can keep them for quite a time. And when we are using the fibrotic, profibrotic effect, we actually can see the increase in the markers. And then we also see it in our fluorescence imaging that we do have increase in the collagen productions, and we also have it numerically visible or different fibrotic markers. We have also we have also done a treatment with the antifibrotic drug, and you can see that this effect is actually reversible when we use the drug. Then yet another method you can use in our system is actually using the 3D printing. Here is actually a part of our Eurostar project where we use a volumetric 3D printing. So it's like contactless printing inside the reactor using the holographic projections. So we can actually print objects like those shows here. This one is made for studying the flows or a growth of the cells in those constructs. And yes, we have grown the cells. We also checked are they live or in the construct, how long they survive. And we use it for the assessment of an more design construct where we wanted to have immediately structures close to the tissue which we are studying. Here I can also show you that this is not so long published paper from Giuseppe Saioli, and we were showing that we can actually use this stable fluidic system to create a very nice and very reproducible neuronal constructs. And very similar work is actually shown by Oscar Kyang. And here I would like to basically show the comparison between two methods. And I am very much always showing this, there's two pictures where basically you have the same type of cells in both systems. However, in the gentle system, the cells are able to organize much easier than on the shaker, and you have a much more rosettes created in the system. And yes, we also can see differences in the conditions where we use then oxygen, when we control the oxygen level. When you control the oxygen level, you can see a big differences in the stiffness of the construct. The yellow thing which you see here is actually those are actually actin fibers. So systems grown in the standard conditions, so 18% of oxygen, like a standard incubator, are much more stiff than the ones grown in the hypoxic or physioxic conditions. And that has an effect on, of course, on the cellular mobility and drug metabolism. We also use our systems for differentiation of the IPS-based liver models. So that one is also a very interesting application where we were able to use the system for whole propagation and differentiation of the IPSs to a normal liver-like constructs. And just to close to the finish, I would like to show you also that those constructs, which are quite sizable, are also have been used as a host for the virus. So that's one of the studies where Rommel Stewart have used the construct instead of experimental animal for an infection studies where those cells, when they were growing in the monolayers, they were not able to accept the virus, or virus was not infecting them. In the construct form, the virus was able to infect those cells. And then they studied actually two different types of COVID viruses, and then you can see the retention differences in the retention in the neuronal tissue between the BA5 and the original BA1 virus. And one of the our not so we just published a paper about using our systems for preparing a material for xenotransplantations. We always often say, okay, 3D methods are replacing or reducing animal use. We are also saying they are very much refining or animal use, where we were using our construct to actually do the xenotransplantation into the mouse. In this way, we basically could use more than 95% of our animals to the end experiments, when we use direct and monocultures, we have a much smaller percentage of usable animals. And just for the end, I would like to show you this nice study published in Cell Stem Cell, made in John Wu laboratory, where they actually were creating an blastoid. So this is actually a artificial construct made out of induced pluripotent stem cells, and it actually mimics the blastocyst. So pre-embryonic construct. Those constructs are coming from the bovine cells, but you basically see that those are very similar to the original blastocysts, and they have been made from the induced pluripotent stem cells, so without any reproductive cells at all. And if you would have this construct kept a bit longer in the system, this is what you can see. So we have, let's say, a pre-embryonic development in this system. So just to conclude, that ClinoStar is actually an environmental chamber, let's say like this, which is able to sustain many different conditions, and we can regulate environment according to the tissue we are modeling. So with that, I would like to say thank you very much for your attention. And if you have any questions, I will be very happy to take them and answer. Thank you.
Thank you very much, Krzysztof, for this very nice presentation. Are there any questions?
Yeah, I can see one hand.
Yes. Okay, George, go ahead.
Hi, Krzysztof. Thank you very much
Hello.
for your wonderful talk. Really nice system. To be honest, I was not aware, and I should have been. So I'm just curious about one technical detail, which is the starting point. So you have a need that your organoids or spheroids in the beginning already have some homogeneous distribution in the space, because if they don't move a lot, then you have areas where you have much higher density of your 3D cell cultures, and others that will be more sparse. So do you have any comments on this?
Yes, I think I should show you something. This system is using a physics to come one, yes, to distribute the objects. So now you don't have to have an already this is a liquid. So you will just see now in this movie. Oh, I didn't share it. Did I share? Now I would share it. You will see it how actually it's look in the lab. That's actually moving from our lab, and then they will go down basically, and then when they start to move, they will be redistributed by the system. So well, Kira took it out, and then you will see that they basically fall. So the movement keeps them in the separation.
Okay. And how much compatible is these cartridges with imaging?
They are. That's a very nice question, because yes, they are. They are actually. We have an applications where basically you can use the fluorescence directly inside of this chamber. So people are not taking it out. You don't need to take the objects out. And that's also a very nice thing when you have an hypoxic system, because when you have a hypoxic system, this chamber is actually preserving the hypoxia. So if you will set up the completely almost anoxic conditions, like for the inner cancer, so it's like 0.05% of oxygen in the system, you will have around 15 minutes when you take out the cartridge and put it under your microscope, and you don't have to have a hypoxic chamber to actually do the imaging. As long as you do not open it, it will be it will preserve. When we work with the more physiological levels, like 6% of oxygen, we have like 45 minutes for imaging.
Okay. And do you already have any of these chambers with basically with two chambers instead of one? Like if you want to have like well, most of the microfluidic systems that you have for organ-on-chip.
So first you have to think, okay, why you would like to have those two chambers? What you want to do? Would you like to have?
Yeah, my idea was my idea was
Mhm.
to have a 2D cell culture in one side, and then on the other side you will have the organoids.
So
But you have
on
a
one
permeable
side,
membrane that you can have, things passing from one side to the other.
we do. We don't do that that way. However, if I can, we do have an insert system. So if you have, I don't know if you can see me now. So if
Yes.
this is the cartridge, yes.
Mm-hmm.
So first, if you would like to have a different cell type, we will put basically different cell types in different constructs, in different organoids. So in different, those are here are actually monoculture. There's only hepatic cells, but you can actually mix them. And because we have this labeling, we can easily distinguish them. If you would like to have this two chamber, and for example, you would like to have an air liquid interface, you will add something like, I don't know if I can show it here. We have such an inserts which you put inside of it. So, and then you can actually have the air liquid interface.
Okay.
But and if you want to have two chambers, because you would like to have a one where you have an let's say liver cells, and another one where you have an cardiacs, because you would like to have the a drug which is metabolized by liver, and then you can check. You want to check what is on the what is happening with the cardiacs. We will suggest to actually do this into a different in the different chambers. So actually collect the media and put them into the different chamber. That's a more controllable system.
Thank you for the suggestion.
Mm-hmm.
Okay, thank you, George. Another question from Michelle.
Yeah, thank you very much, Krzysztof. Very nice talk. Just a curiosity, you show those chambers and it has those blue spheres in it on the side. What are those spheres?
Yes.
Yeah, that one.
So if what you see here, this chamber is like here we have a cell chamber, and here you have a media. We actually don't use a phenyl red from different purpose actually for imaging, but that blue balls they are actually a water. It's a solid. It's a polyacrylamide based beads which actually absorb water. And what they do when this thing is rotating, it actually has some inlets for the air. So air goes into it and goes between. This is like a lung. It's actually moisturize the air and prevents evaporation from this chamber, because we do not want to have evaporation from it.
Sure, sure.
Okay. And then it also gives you this heat, because it's if you have it like this, then it preserves the heat. So when you take it out for the imaging, it also does. We did this because we didn't want to have an open water in our incubator, because that's a potential infection. And then each of them has exactly the same humidification. So you don't have this problem that, let's say, okay, my middle shelf in the incubator is great, but my bottom or top are not nice, yes? So we don't have that.
You can dose there as well. You can make some with some dosing of compounds, or that
you are actually touching a thing which DARPA want to do. Yes, because if you want to have a gas or the airborne pollutants, yes, that's the thing. Yes.
Okay. Thank you.
Okay. Thank you, Krzysztof. We should move on to our next speaker. Thank you very much again. We now move to our next speaker, which is Carina Valente. And Carina is a researcher at the Laboratory of Molecular Microbiology of Human Pathogens. Her research interests are focused on different aspects of Streptococcus pneumoniae biofilm colonization of the human nasopharynx and how these impact on progression to disease. Her research has been dedicated to understand how pneumococcal strains coexist and interact in the biofilm environment, and how dissecting the molecular mechanism governing those interactions. Currently, she is working on the development of a more physiological model to study Streptococcus pneumoniae biofilm colonization and its interaction in the human host. And today she's going to talk about development of a nasopharynx-on-chip model to study Streptococcus pneumoniae colonization. Thank you, Carina, for having accepted our invitation, and you can start sharing your screen.
Okay, I'm sorry. Should be on the first slide. Sorry for this. Okay, now are you seeing my screen?
Yes, you can start.
Okay. So good afternoon to everyone, and thank you for the invitation to speak about the project. So my first I would like to say that the project, so we've only started implementing the project now, so the first experiments are ongoing. So I don't have actually results to show you, but I will guide you through the project goals and the strategy that we are going to use to implement the model. Under the scope of this presentation and the scope of the project, I will also talk about my staff exchange, the opportunity that I had to visit the Mosaic Lab in Jena under the scope of the staff exchange grant that I got, and how that experience is helping me in developing and implementing this model. And now I cannot move my slides. Okay, sorry. So first I will introduce you to Streptococcus pneumoniae, which is the bacteria that we study in the lab. So it is a gram-positive bacterium that is surrounded by this polysaccharide capsule that you see here. The capsule is the main virulence factor and is highly diverse in terms of composition and structure, and there are in fact over 100 capsular types or serotypes described so far. Streptococcus pneumoniae is a pathogen. It has been classified as a priority pathogen by the World Health Organization. And the most recent estimates on the death toll caused by invasive pneumococcal disease mentions numbers close to 500,000 deaths per year. And within these deaths, over half of them occur within children, younger than 5 years of age. So disease can take many forms, and it ranges from mild conditions such as otitis media to more severe ones like pneumonia, bacteremia, and meningitis. Disease is treated with antibiotics. Resistant is not, it's becoming a problem, but is not a severe problem so far. And pneumococcal, and disease is also prevented by the use of pneumococcal conjugate vaccines, which have the problem of having limited valency because they target the capsule. And so the vaccines that we have until now cover only up to 21 serotypes, and all the other types are not covered by the vaccines. So from an evolutionary perspective, disease is considered to be a dead end because the bacteria either kills the host or is killed by him and does not transmit to other hosts. And that's where colonization is important. So besides being a pathogen, pneumococcus is mainly a colonizer of the upper respiratory tract, and particularly the nasopharynx. So colonization is much more frequent than disease, and it takes place. It occurs in the form of these multicellular communities called biofilms that are attached to the epithelial mucosa. Colonization is very important because it is a key step for disease to occur. It's during colonization that the bacteria transmits to other hosts, and it's also during colonization that the bacteria evolves by horizontal gene transfer within this biofilm environment. So in our lab, we have been studying pneumococcal colonization in different aspects of this process. So here I have an overview of the events that happen during pneumococcal colonization, and I will guide you through these different steps. So for the initial stages of colonization and the initial steps of biofilm formation implies that the bacteria have to cross the mucous barrier of the epithelial mucosa and attach to the cell surfaces. In this process, the capsule, the negative charge of the capsule plays an important role to avoid the mucous barrier, but also pneumococcal glycosidases, which will cleave the mucus and expose the cell surface receptors of the host cells. At the same time, there is also downregulation of the capsule to expose surface proteins in the pneumococcal surface, and which will then allow that can bind to the host receptors. Once this initial adhesion is established, the pneumococcus will undergo a process of cell proliferation and matrix maturation within this biofilm. But unlike other bacterial species that have systems dedicated to matrix production, in the pneumococcus, there are no such systems, and the formation of the biofilm matrix results from a balance between cell proliferation and cell lysis, because the matrix is composed of polysaccharides and external DNA and proteins that are released during cell lysis. For this process, the contribution of autolysins and other hydrolases is very important, and also cell-cell communication systems that occur within that are present in pneumococcus, and that ultimately result in the death of a part of the population to release those constituents of the matrix. Once the biofilm is mature, some cells will start dispersing from the biofilm. The process by which this occurs is not very well known, but we know that temperature seems to be, studies have shown that increased temperature is a trigger for this dispersion, and also capsule upregulation of some cells also triggers this dispersion. This is very important because it's these cells that disperse from the biofilm that transmit to other hosts, and also it has been shown that these dispersed cells have a higher invasive disease potential than the ones that are attached to the biofilm. Another aspect that occurs, another process that occurs during colonization, and that is important, is pneumococcal clearance from the nasopharynx. So for this process, there are at least 4 mechanisms involved, and there are probably more. One of them is this, the mucociliary barrier. So cilia will beat and will entrap bacteria in the mucus and will promote its release from the mucosa. There is also a role of the innate immune response that is triggered by the recognition of the bacteria by the host cells. There is a role for the adaptive immune response, both humoral and cellular immunity. And of course, there is an important role for competition with microbiota, either by creating colonization resistance or because some of these microbiota members can target pneumococcus directly by producing, for instance, antimicrobial peptides. So this is what happens in the short course on pneumococcal colonization. So the human nasopharynx lining is constituted by this pseudostratified mucociliary epithelium that has ciliated cells, that has goblet cells, which are cells that produce the mucus, and among other cells, there are also basal cells that will then differentiate into the other types of cells. Another important aspect, particularly for bacterial infections, is the mucus that lanes this epithelium, and that is very important for bacterial defenses. There are many models that have been used to study pneumococcal colonization, all of which, of course, have their value and are very valuable, in fact, but none of them replicates the complexity of the human nasopharynx. So abiotic surfaces have been used extensively to study mostly aspects related to the bacteria because they lack host component, and in colonization studies, this is a big limitation. And to overcome this limitation, some models have been developed using immortalized cell lines, but this is one cell type that does not reflect the complexity of the cellular responses. And the cellular responses are not the same. Murine models are also very valuable, but mouse have different host responses, of course. These are different species, besides the fact that S. pneumonia is a strict human colonizer, and establishing colonization with some lineages can be challenging. And of course, we cannot ignore the use of human models, which are being used to study pneumococcal colonization, and that they are very useful to study immune responses, both systemic and mucosal immune responses in response to colonization. But with these models, we cannot capture and we cannot evaluate direct cell-cell interactions, and we cannot evaluate the dynamic of biofilm formation. And this is where our project comes. So clearly we need a more physiological model that we can use to study Streptococcus pneumonia colonization. And that's exactly the aim of this project. So we have different research questions in our lab, and which we would like to use this model, but specifically we want to study mechanisms of biofilm colonization, the cellular and the molecular mechanisms of biofilm colonization. We want to understand or to evaluate both the host and the pathogen responses when cells are interacting with each other. And there are also other research lines in the lab that could take benefit from such a model, because there's a research line that is addressing the development of novel therapies that target colonization as a means to prevent disease, and to study as well co-infections, either with viruses or other bacteria of the same and different species. So the project within the MPS NOVA, the project has 2 tasks and 2 two specific objectives. One is to optimize the nasopharyngeal on-chip model, and the second one is to establish the model of bacterial colonization in that nasopharynx-on-chip model. So for that, we will use nasopharyngeal primary cells, monocytes, and endothelial cells. And by these cells, you might ask, so our main questions our main research questions are depend on epithelial responses. So we will use primary cells to be differentiated in a mucociliary respiratory epithelium to have the complexity of the nasopharynx. Monocytes, we will use the THP1 cell line. So we will use monocytes to be differentiated into tissue-resident macrophages, which are important for phagocytosis and for conditioning epithelial cell responses. And finally, we will also use endothelial cells, despite the fact that we are more interested in the epithelial response. Studies have shown that the crosstalk between endothelial and epithelial cells promotes a more robust mucociliary differentiation than using only the respiratory cells alone. So for this purpose, we will also use endothelial cells. So under the scope of the MPS NOVA, I was fortunate to have the opportunity to visit the MOSIG lab at Vienna University to have hands-on training on organ-on-chip development. So during those 2 weeks in which I was there, we worked on a lung-on-chip model, which is very different from the model that I am proposing to develop. But at the same time, Ristina Kocheva, which is here lighted with the yellow circle, is working on she was working on a bronchi model derived from human pluripotent stem cells, which is more similar to the one that I am proposing to develop here. And so I had the opportunity to benefit from her experience and to squeeze all the information from her that I could at that time. So this is the model for lung-on-chip. So the chips have two cavities in the model. In the lung model, the upper cavity was used to represent the vascular side, and the lower cavity was used to represent the epithelial side, so the lung side. The first days of the model are for cell seeding, and the cells are cultivated in static conditions. And by day 8, they are put in perfusion. And for the complete differentiation of the lung cells, they need to be exposed to air, to be in an air-liquid interface, which happened by day 12. And by day 14, the model was ready for whatever we wanted to do with it, which in our case was to test infection. Here are representative images of different stages of the process of developing a lung-on-chip model. So here we have an image of the chip at day 4, the vascular site, in which we were checking for UVEC confluency before seeding the epithelial cells. So during the chip development, we check before every news process, we check the state of we visually inspect the chip to control for good development and confluency. And here is the final result at day 14, my not so perfect chip, but it was the first one I did, so I was very happy with it. In which we have epithelial, the epithelial layer. And we see the cell membrane of epithelial cells in yellow, macrophages, which we almost don't see, but stained in red, and the nuclei of the epithelial cells stained in blue. So, but this model is very different from compared to the one that I'm planning to develop. Still, the aspect, the learnings that I obtained, or the knowledge that I obtained there was very, very useful to transpose for the development of my nasopharynx-on-chip model. In particularly, I would like to highlight these aspects here in this slide. So first, the technical aspects of handling chips. So how to sterilize them, how to all the tricks and tips that are useful. So this was very nice in this. It was very useful in this sense. The technical aspects associated with the flow system, which cells to include in the model and their culture conditions, and which cells not to include in the model, the timings and amounts of seeding and the culture media, the mixtures in culture media, etc., etc. Considerations for infection, specifically multiplicity of infection, timings, etc. The possible readouts of the chip and how to collect samples for each application, and finally considerations for model characterization, particularly by immunofluorescence. So even after everything that I learned at the Mosaic Lab and everything that I read in the literature, I had many doubts. I still have many doubts. And at this stage, I had very important and productive discussions with experts from Dynamic 42, which are the company in which commercialize the chips that I'm going to use. So they were very helpful in the selection of the chips and in some details of the protocol and some aspects that I was not aware of, and this was very, very helpful and very important. And integrating all this, the model that we are proposing to develop and the workflow for the model that we are proposing to develop is summarized here in this figure, in this schematic. So in the upper side of our chip, we will have the epithelial side. So the pseudostratified epithelium with macrophages, and in the lower cavity we will represent the vascular side. So the inversion of these two compared to the lung model, these layers are inverted because the upper cavities are big in terms of volume, are bigger than the lower cavities, and this is useful for the differentiation of the pseudostratified epithelium. One particular and the important difference is that air-liquid interface in this model takes about 21 days. So it's very important that the macrophages and the epithelial cells are only added after complete differentiation of the epithelium. And it's also important to take into account the precautions not to revert differentiation when seeding macrophages, for instance. So every time we see the new cell, we will have to change from perfusion to static conditions for some time, and then resume perfusion once cells are confluent. The same happens for infection at day 31, and it's also important to take precautions not to revert the air-liquid interface. At the end of the experiment, we will have to characterize the chip, the nasopharynx-on-chip, and we will do that by immunofluorescence and by checking barrier integrity. We will use cell markers for each type of cells that we will have, and also markers for tight junctions in the epithelial layer and in the vascular layer. So As I mentioned in the beginning, experiments have just started, so we are doing at the moment we are doing a pilot experiment for isolation and differentiation of human primary nasopharyngeal epithelial cells. So we have identified 4 healthy donors, 2 males and 2 females, and we collected 2 swabs per donor. And basically, what we are doing now is just preparing, growing the maximum capacity these cells with the minimum passages, of course, to prepare stocks and then use a transwell system to differentiate these cells into a pseudostratified epithelium. And to control for reproducibility and for complete differentiation, which we'll evaluate using the same markers that I mentioned previously. So, as a summary of this talk, I hope I have convinced you that this nasopharynx-on-chip model could be a powerful tool for research on pneumococcal colonization. I've guided you to the implementation plan to develop this model, and I have also shown you the invaluable contribution of the staff exchange for the implementation of the project that we are doing now. I hope in the next few months I have results to show you. Finally, I would like to acknowledge the people that are involved in the project, so which is my supervisor, Raquel Salião, and Alexander Mosig at Jena University. The Mosig labs, they were very welcoming when I was there, and Ristina was amazing in everything that she taught me, and she was and is being very useful. MPS NOVA for funding, both for the research grant and the staff exchange grant. And last but not least, the amazing lab where I work at ITQB NOVA, and of course ITQB NOVA, where the project is being developed. And I'm hoping that you have questions and suggestions for me. Thank you.
Thank you, Carina, for the nice presentation and for showing your work or your plan for your work. Is there any questions or suggestions to help Carina?
I have a question. Out of curiosity, because we are also planning to use these THP1 cells, but in our case, so we are implementing a gut-on-chip model. But we were thinking to place this THP1 in the endothelial site, and I was curious to see that in your plan is that you plan to do this in the epithelial site, and I was curious to understand why, and so that will help me to understand why I do it in the in the endothelial site.
Because I want to differentiate these cells into tissue resident into macrophages. So are you differentiating them in macrophages as well?
The idea is to differentiate them as well, but what I understood is that they can migrate through the membrane to the upper side. That's why
they can. So yes, they can. But in this model, it would be difficult because my membrane is 0.4. It's very small, so there will be no possibility of the cells to cross that pore size. But so macrophages are in tissue, so for me it makes more sense to have them on the epithelial side and not in circulation. But yeah, so I need to have the macrophages because the response of the epithelial cells is also conditioned by those macrophages, and that's why I have them on the epithelial side.
Okay. Yeah. Thank you. Yeah. Because I always saw them in the other side, and now I was very curious, but then I will read a bit more about that.
On the lung side, they're on the lung side. On the lung-on-chip, they're also on the epithelial side.
So notice, yeah. Okay, thank you.
Thank you. Is there any more questions or suggestions? Okay. I don't think so. So thank you again, Carina, for accepting our invitation. Thank you again to both speakers. It was a really nice seminar, and I hope to see you all in our next seminar that is in May 7. And thank you all again.
Thank you.
Bye.
Bye.
Bye. Thank you.
Thank you.